So in this last lecture for Chapter 14, we're going to take a look at the factors that cause blood type. So there's something on the order of 31 genes that are known to control the production of identifier proteins or molecules that are found on the surface of red blood cell membranes. These identifier molecules may be proteins or glycoproteins, but they're all used by the body to, as a means. Identifier proteins like this are used as a means to ensure that every cell that a white blood cell encounters is one that belongs in the body. So if it has all those self-identifying, chemicals that are found on the surface of the membrane, then the body will recognize that cell as self. And there are two surface antigens. They're glycoprotein A and glycoprotein B that are produced by a single gene or at a single gene. And so we have three alleles or three versions of that gene that can be found. There's the A version, there's the B version, and then there is a non-functional O version. Then there's a second gene that's called the Rh gene. And so whether or, how or what surface protein is found on the surface of the cell then is a matter of heredity. So if a person is type A, they would have these A antigens found coming off of the surface of the cell. If a person is type B, then they would have the B antigen. If they're type AB, then they have both the red antigen and, the antigen A and the antigen B found on the cell. Different locations, but it's got both of them. If, in the final instance, they are type O, that is a person who doesn't have any functional surface antigen from the ABO group found represented in the body. So, blood type, it really describes the antigens found on the red blood cell's surface membrane. If a person is type A, they have the A antigen. Type B, they have the B antigen. Type AB, they have both antigens on their cell membrane. Type O has no ABO or no AB antigens on their surface membrane. There is then two versions of the gene for Rh. Rh positive has a functional Rh antigen. Rh negative doesn't have that antigen. So if we were to look at somebody's heredity, they may have a gene for A, a gene that codes for B, and then at a second site on the DNA, we would see this Rh antigen, if they're Rh positive, or no functional antigen. So AB positive is what this cell would be. This would be an A. It had an antigen, but no Rh antigens. Then it would be A negative. So also in the blood, so we have the red blood cells in the blood, but also found in the plasma. So now this is not on the red blood cell surfaces at all, but in the plasma we find proteins. They're the globulins that are in the blood plasma, and they are there and prepared to take out any foreign cells that don't belong, or to assist in taking out any foreign cells that don't belong in the body. So these globulins, and here we can see the globular proteins, they look a little bit like Ys. They will bind to identifier proteins that do not match the genes of the of a self body. So these globulins are also referred to as antibodies. And so a person who is type A, that means they have the A antigen, well then they will produce globulins that help to remove anything other than the A antigen away. If it's type B, then there are B antigens on the red blood cell, and in the blood plasma, we would find anti-A antibodies. If a person is type AB, then we would expect that they would have neither the anti-A or the anti-B. Oops. A person who's type O, because they don't have any of the antigens, they're going to have anti-A and anti-B antibodies. Okay, so that's for the ABO group. Oops, sorry. Everything up above the second line there. That is the ABO group. And then if a person is Rh positive, they have the Rh antigen, then they will have no antibodies for the Rh group in the blood plasma. And if a person is Rh negative, then we would expect to see that they would have anti-Rh antibodies in the blood. So if you've got the so if you have, let's say, the Rh antigen, you definitely wouldn't want to be attacked by antibodies that would take out the cell and kill it, because it is there identifying, marking up that cell as being a self body, a self cell. So cross reactions can occur as blood cells that have surface antigens on them that are incompatible with the antibodies that are found in the plasma are introduced. So, for instance, a type B person, if you're type B, then you have anti-A antibodies. If you're type B. And if you deliver to someone with these type B antibody or anti-A antibodies, so here's anti-A antibodies all over the place, then you introduce a cell in there that is a type A cell. And remember, what kind of antibodies were these? They were antibodies from a type B person. So those antibodies are going to stick to that A and signal to the immune system to come and destroy. Now, because a single antibody can bind two different substrates, well, that just means that the single antibody here, there's the antibody, it combined a antigen here, and over here it can also bind an A antigen. So that helps to that causes, I guess that's the bottom line, is that this antibody here, because it combined two different cells, then it's going to cause clumping. It's going to cause the aggregation of the type A, in this case, clumps like we see here. So there is a bunch of aggregated, or agglutinated is the word that we use, agglutinated red blood cells. And these red blood cells may wind up lodging in a blood vessel, which can be really, really painful or potentially fatal.